of Research in Biomedical Engineering, and Dr. Glenn Gaudette's team at the Worcester Polytechnic Institute recently made headlines after they demonstrated use of the common spinach leaf as a vascular system in cardiac medicine. Just how did they grow muscle cells on a plant scaffold, and can their methods be applied towards cultured meat? Let's find out. Please join me in welcoming Dr. Glenn Gaudette to the stage.
Thank you. Well, thank you very much for the opportunity to speak to you today, and thank you for all the work that's going on in the room. I come from a medical background, so our first slide is generally our disclosure. You know, do I have any companies that are giving me money that might be related to this? The answer to that is no, but my disclosure is my background's in tissue engineering, and I don't know a lot about the clean meat, the cultured meat industry, but I'm happy to say that I'm learning a lot, and wow, what an exciting opportunity, an exciting time to be alive in working in this area. So I've learned a lot from New Harvest. I've learned a lot from Marianne Ellis, who's here somewhere, and other literature that's out there in this nice book on clean meat. I remember about 6 months ago talking to Marianne Ellis, and she shared with me some of her work. And it's been very interesting to me. And so hopefully, as the talk goes on, you'll see some of the potential opportunities for the work we're doing in this field here. So our focus is on tissue engineering. Why? Why do we care about tissue engineering? The bottom line is there's a huge organ shortage out there, which I'm sure you're probably aware of. And so this pink line here, the difference between the pink and the blue is the number of organs that are needed, and the blue line represents the numbers of organs available. And so there's a huge mismatch there, and we need to see if we can decrease this gap. And so tissue engineering hopefully can take place and help address that gap. So what are we doing in the field? We've taken fibrin sutures, and in the upper left-hand corner there, you can see that suture actually contracting. And so that's made of fibrin. Fibrin's a protein found in blood clots. We've coated it with human pluripotent stem cells that have been turned into cardiac myocytes, so cardiac muscle cells, and that contracts. On the bottom here, what we've done is we've labeled these cells with a fluorescent dye, essentially, and that dye glows, so it turns white if the calcium inside the cell goes up. And so if the electrical activity is present inside the cell, it'll go up. And so what you see when that movie's playing is you see the white, the brighter cells. That means that they have electrical function. And then in one more video I'll show you here is a close-up of some of the mechanical contraction that also occurs. And so in the heart muscle, we have mechanical and electrical function going on. So we can get these cells, we can take them, we can turn them into heart muscle cells, we can get them to contract on this fibrin suture, which is approximately 100 microns thick. But the big problem becomes once you try to stack these here on top of each other, there we go, and make a thick scaffold, a thick tissue graft, we have a diffusion limit. And so we heard a little bit about that in the previous talk. And so what is that limit? That limit's about 200 microns, right? And if cells get outside of that limit, they're going to die, unfortunately. They cannot get enough oxygen. So we can take these sutures, we can get them to contract, we can stack them on top of each other, but if we want to keep the cells alive, we need some sort of vascular network. And so we started looking at current biofabrication techniques to see if there's anything out there that we could use. And our colleague down the road here at Mass General Hospital, Harold Ott, decellularized rat hearts. And so what you see up here on the upper right is an actual rat heart. And on the left is with the cells, and on the right is without the cells. And so some folks refer to that as a ghost heart. And so Harold was able to remove those cells by perfusing that heart with detergents, soap essentially. The soap breaks down that phospholipid bilayer in the cell, and then the intracellular contents are allowed to escape, and we just slowly wash it out. Right? And so he did some studies to look at what materials left behind and all that there. But the interesting thing here is that vascular network stays in place. And so we thought this here was an interesting opportunity. We worked with Harold and actually done the same thing with human hearts. And so we can decellularize a human heart. But as I mentioned before, there's a big mismatch between the number of organs needed, the number of hearts needed, and the supply. And so a big supply and demand issue. In addition, these tissues are expensive. and they're variable from one patient to the next. So we wanted to see if we could find a more sustainable, a more reliable source of tissue to grow cells on. And of course, where else to look except at your salad and at spinach? And that's what my graduate student actually did one day at lunch. There was a piece of spinach on the table, and we took a good look at it. And if you look closely at that slide, hopefully you can see those veins that are in that spinach leaf. And when you think about the mammalian heart, and again on the left is the rat heart, you can see the branching vascular network in the rat heart, and you compare it to the plant leaf on the right, which has been decellularized, and the veins have been loaded with a fluorescent dye that you see in green. Hopefully, the similar vascular structure jumps out at you. And that's what happened with us at lunch. We said, gee, this is pretty interesting. And of course, we work in a tissue engineering lab. You're not allowed to have food in the lab and all that stuff there. So, Josh is a very sneaky guy, so he was able to do it in the back of the lab where nobody saw. Question we get quite frequently, well, where did you get the spinach? Well, literally, we went down when I say we, that means my graduate students, of course. So we went down the road to Subway Sub Shop, and Josh asked for a handful of spinach, and they looked at him strangely, but they gave it to him. And so then he brought that back to the lab, and now we go, we really stepped it up. We go to the supermarket now to get our spinach, and we have to write not food on our containers because it's not actually food. So, but anyways, we looked and we said, okay, can we do what we do with the rat heart to the spinach leaf? And so what you see here is a rat heart being hung by the aorta. And so the tube coming down from the top is actually a tube with detergent in it, and it perfuses down the aorta through the coronary arteries, and that's the way we decellularize hearts. When you look at the end of a spinach leaf, and hopefully, and I guess I should have warned you, you may not look at spinach the same way after this talk. So if you look at that spinach, you'll see a nice hollow tube, essentially, in that leaf, right, in the stem. And so Josh said, gee, this is perfect. This looks just like an aorta to me, right? That's what we do in medicine, right, is everything looks like a heart tissue or something like that to us. And so it looked like aorta. He stuck a cannula in there and actually was able to attach that to a bottle that has detergent in it, the same detergent we use to decellularize rat hearts. And so we start off with a piece of spinach like this. We perfuse it with SDS. This is a detergent. And originally, we perfused it for about 5 days. And so what's happening here, again, is that detergent is breaking up that cell membrane, and all the cell contents are now being washed away, right, over 5 days. And then after that, we switched detergents. We used Triton X, and we also add some bleach in there. We like to do things in a sterile environment, and plants are not really that sterile, and so we want to make sure that if there are any bugs inside of that plant that we kill it, and also help wash out any more material that might exist there. And so the big question is, are those veins still open? Right? And so what we did here is we perfused red dye down the needle that we stuck in the top of the spinach leaf. And hopefully what you can see here is the red dye being perfusing that leaf. And so I have a close-up on the right-hand side of this slide, and you can see that red dye going throughout that clear leaf, right? Suggesting that we're able to perfuse that leaf and those veins are still open. Right? And so we then asked the question, well, that's great. It looked great. And that was my colleague, Tanya Dominko, who's sitting right back there. That was her great idea to perfuse it to make sure that we were able to show that the vessels were still open. But we wanted to go a little further. And so we perfused it with different size microspheres. And what you can see here is this white dot going across the screen. That's a 10 micron diameter microparticle, microsphere. And why do we choose 10 microns? That's about the size of a red blood cell. And we wanted to make sure that those particles could get through the leaf. And so what you see here is that a pseudo-colored of a particle actually going through the vein of the spinach leaf. We looked at a bunch of different size particles. And so our capillaries are about 10 microns roughly in diameter. And so if we were to perfuse muscle tissue, normal a rat heart, with 50 micron diameter spheres, they would get stuck because they wouldn't be able to go through those capillaries, right? Well, we wanted to see if we could do the same thing with the spinach leaf. And so what you see is when we perfuse the leaf with 50 and 100 micron diameter microspheres, about 50 and about 15% of those microspheres are actually able to get through the leaf. Most of them get stuck in the leaf, and that's what you see in the bottom right-hand side there. But when we perfuse it with 1 micron and 10 micron diameter beads, those beads go right through that vascular network in the spinach leaf. We've actually recently been able to perfuse it with blood. And so this here is actually whole bovine blood that we perfuse the spinach leaf with. And so on the left, you can see the red blood inside the vein. And then if you can actually click on this movie here on the right-hand side, we cut part of the leaf, and we're still perfusing it with blood. And you'll see the blood come out in this beaker of water here. Hopefully that plays. Can you click on the right one here? Yeah, thank you. And so there you are. You can see the blood coming out from the top. Again, we cut one of the veins on the leaf to show that it would come through there. And also on the bottom, you can see the blood coming out through the bottom of that leaf, and that's in a beaker of water. So we're able to show that we can actually perfuse these spinach leaves now with whole blood. So we feel pretty good. We're able to perfuse them. We did some DNA work, and again, I heard about collaboration. Collaboration is so important. We're fortunate to have a lot of great collaborators right at WPI, but also at other universities. And so Tiny Dominko's lab looked and made sure that there was no plant DNA left. We showed that fluid can go through the vasculature. We showed that blood cells should be able to flow through the vessels. But the big question for us is, can we actually get human cells to adhere to this? And so the first step is to coat it with endothelial cells. And so our blood vessels are coated with endothelial cells. And so we actually injected endothelial cells. We labeled them red. We injected them into the main stem of the spinach leaf. And what you see here are the cells lining the inside of the vein of the spinach leaf. And those are human umbilical vein endothelial cells. We also looked at human mesenchymal stem cells. And so we've done some work also with HMSCs. And we were able to seed these HMSCs also on the outside of the leaf. And we labeled these here with a different red marker, but also a red marker. And in the background, in the middle slide there, the middle panel, you can see the actual veins in the spinach leaf. The big question, though, is can we get human heart muscle cells on there? And will they actually function and contract? And so if you look within that yellow circle, you can see that cluster of cells actually contracting. And then if you look on the right-hand slide, if you can play the right-hand panel, if you can play that movie there, yeah, perfect. You'll see the bright white signal again, suggesting that there's electrical activity in these cells that are now coating the outside of this leaf. And so these are human cells. These are induced pluripotent stem cells that were then turned into cardiac myocytes, human heart muscle cells, and they can actually contract and function on that leaf. There. And what are some of the other opportunities? Well, our colleagues are interested in bone and regenerating bone, and so we've started looking at bamboo. And we've asked the question, can we use bamboo? Can we use other plants? Are there other sources available? And if you think about all the different form factors that plants come in, and all the different mechanical properties that plants have. And so our colleagues have said at University of Wisconsin and also at WPI, can we use bamboo? And so we've decellularized bamboo, and you can see in the top right, that's a cross-section of decellularized bamboo. So we remove the cells from the bamboo. Our colleagues can also biofunctionalize it, and so they can coat it with minerals, with calcium, for example. They could coat it with RGD peptides to increase cell binding. And so earlier we had a question, will the cells bind to the leaf if it's coated or if it's uncoated? And the answer is yes. They bind to the leaf when it's uncoated. Do they bind better when we coat it with RGD peptides and things like that there? Yes. But they will certainly bind to the uncoated leaf. And we've also started looking at broccoli. And so if you think about lung and the structure of lung, that also, to us, looks like broccoli, right? I know you guys are probably thinking of something else, but it looks like it looks like lung to us. And of course, it's getting close to lunchtime, so we'd probably rather real broccoli. But one of the things I want to point out in the middle panel is we haven't perfected the technique yet with broccoli. And so you can see some of the dark spots in the center there, and that's because we're not fully decellularizing the broccoli. And so we still have a little bit of work to do over there. What are some of the additional benefits? It's a green technology. When we think about tissue engineering, unfortunately, a lot of the scaffolds we use, they're really not good for the environment. And so they cost a lot of money. And what are we going to do with all these this material that we're going to waste? So hopefully using something like plants and plant-based technology will help move the medical industry into a greener area. And one of the exciting things to me about the work that you all are doing here is the significant contribution you can make to our environment. And to me, it's just been so exciting. Judah Folkman, who's known as the father of angiogenesis, was passed away, unfortunately, a few years ago. He was at Boston Children's Hospital. I remember talking to him once, and he said, there are two types of research. There's a research that pays the bills, and I'm very fortunate to have some research that pays the bills. And then there's the research that wakes you up and gets you up in the morning that you're excited about. And I'll tell you, when I started hearing about the work that you folks in this room are doing, that is now what gets me up in the morning. Just being able to think about the effect, the positive impact that you folks can have on the environment is really exciting to me, and it helps get me up in the morning and gets me excited. Plants can be grown in many different shapes, many different forms. They can be genetically engineered. You can express different proteins in the cell wall. And so when we remove the cells, we remove the cell membrane, and we remove the cell, but the cell wall stays behind. And so we're working with colleagues at Arkansas State University. They're able to transfect these plants with all kind of proteins, and the proteins will stay behind after we've decellularized it. Plants can be grown in good agricultural practices, GAP. I had never heard of that before. I'm familiar with good manufacturing processes. I'm familiar with GLPs, good laboratory practices. But I became familiar with GAPs, which are very interesting. Plants are abundant and readily available, and hopefully they can bring the cost of medicine down. So where do we go? Where do we go next? I go right back where I started. I had a great conversation with Mary Ann Ellis and her colleague yesterday out at WPI. I look forward to more discussions. I look forward to seeing if there's any way we can help you folks try to do what you're doing and continue that good work. Again, I'm honored to be here today, and I thank New Harvest for reaching out to me and asking me to present here today. And the work they're doing is phenomenal and fantastic. And some of the papers in the industry, they're fantastic. And I just quickly want to mention this blog here in the middle, Making Steak Out of Spinach: How Bioengineering Can Change Meat Production. That just appeared one day. I had no idea about that, and some student down the road here at Harvard wrote this, and I was like, wow, this is really pretty neat. It wasn't my idea to grow meat on spinach, but it came from a community that is open and willing to share and help each other out. And to me, that's the way science needs to be in the future. And just keep the books coming. I enjoy reading about the field. So. To summarize, none of this work, and just to wind down, none of this work can be done alone. We're fortunate to have many graduate students and undergraduate students and even high school students working on this research. And I'm fortunate to have Mark Bray and Logan Gaudette with me today, who've done part of this work here in the lab. I've listed my colleagues here. Tanya D'Amico is also here. You want to pick her brain. She's fantastic too. Pam Weathers is a plant expert at WPI. And 3 years ago, I did not know the difference between phloem and xylem. It'd been a while since I had that biology. And Pam helped me understand that. Jeanne Coburn was down the road here in David Kaplan's lab doing a postdoc there, and she's a colleague now at WPI and doing great work. University of Wisconsin-Madison, Bill Murphy is fantastic, a great person to work with. Arkansas State University, they're doing a lot of the plant work and helping us get a better understanding of plants. Mass General Hospital, Jeremy is a plastic surgeon, a burn surgeon who wants to use this here to treat skin. And of course, Mary Ann Ellis, who I did spot back there at the University of Bath, a tremendous help too. So happy to take any of your questions now. Thank you.